My name is Amin, and I'm going to present my poster on neural data analysis pipelines for extracting information from C. elegans microscopy data. Recently, a biological technique introduced a differential coloring scheme in the C. elegans, where you can introduce different protein fluorescent protein markers into different neurons, and you can color them in a deterministic manner. So this is an example of how this data looks like. So we have different neurons in the C. elegans that are appeared in different colors, and then many of the biological questions that are asked and answered right now, they require some sort of manual work where you have to go through these images. These are volumetric images. You have to determine, you have to identify the cells, and at some regions in the image, identifying the cells is going to be a bit harder. So we should kind of incorporate information both from the spatial posture of the worm as well as the colors of the neurons to determine where the neurons are. And then once we determine the identity of the neurons, then we also have videos and together with this biological technique, which is called NeuroPal, we can also express GCamp in the worms, and then can record the activity of the neurons as the worms are moving and behaving. So what we did was to grab these data sets and develop some pre-processing pipelines to extract information from these videos. Namely, we actually developed multiple methods that I'm listing here. So the first one is atlas construction. So we know that the worm body and worm neurons are kind of a stereotypical. So we can go ahead and use this as stereotypical stereotypy to kind of develop a statistical atlas. So this is how the atlas looks like. So we are assuming that there is a latent space where every neuron has a center and it has some sort of covariance matrix. And then different worms are actually draws from this distribution. So we can try to incorporate all the information from multiple worms to develop a statistical atlas of neuronal positions and colors. And then what we can do is that we can, for a new image, we can try to register this new image to the atlas that we have developed. So this is what we did. So there is another work where we can grab this atlas, which is this red point cloud, and grab the neurons in the image, which is this green point cloud, and try to match these neurons, align these neurons together. Once we do this, we have the aligned space, but then we have to determine, based on the neurons in the atlas, we have to assign an identity to every neuron in this image. So for that, we used a method that is called a Sinkhorn's algorithm. So it gives us some sort of probabilistic assignment of the neurons. So we can say that neuron that is appeared in a certain position is 50%, has 50% probability to be assigned to neuron A and 50% to neuron B. So this way we can also deal with uncertainty. We furthermore developed a probabilistic segmentation method where starting from this statistical atlas, we can actually write down a Gaussian mixture model with some sort of prior distribution and treat the pixels in the image as Gaussian observations, and we can fit this model and get the segmentation in an unsupervised manner without having training data. And finally, we also did some work in demixing calcium signals in the moving animals. So when the animals are moving, we cannot use traditional matrix factorization models because they're assuming that the tissue is fixed. Since the tissue is moving, we introduced this new functional form, which is we are transforming this canonical space into different time points. So there's some sort of deformation in this model. And yeah, these are basically the multitude of the methods that we have developed for analyzing this data. And many of these methods are also applicable to other model organisms. Finally, I'm listing the papers that we actually published from these methods, and three of them recently published in MICI. And also wanted to note that the code and software are also all available on my GitHub page.
Thank you, Amin, for the nice and brief descriptions of your work. If you have any questions for Amin, please post them in the questions and panel sessions. order through the chat, and we will read them for him. I mean, could you please explore? I mean, is it possible to use this method for other animal models? And
Yes.
if so, what kind of modifications or skills do we need to be able to use it for other models?
Yeah, that's a very good question. So yeah, for sure, many of the models that we developed, they can be applied to other model organisms. In order to develop an atlas for a different model organism, we need to have some sort of a stereotypical aspect of the data. So here we know that the neurons relative to body are appeared in the same position. So if you have that, that's sort of, and this is very much similar to what is done in other data sets, like MRI, for example, where you actually develop some sort of atlas for the brain, for the human brain, which is like an average human brain. So this is what we're actually doing here. So all these steps that we are following here are very similar to what is done for human MRI. Atlas construction, image registration, detection of different regions, and then segmentation, and then like reading fMRI signals, for example. So this is very much in the same spirit. Yeah, so like the atlas depends on the the stereotypical and this other method, which is just the point cloud registration. There's like a rich literature on that. We also did some theoretical work. So if you have like 2 point sets, you want to register them, that depending on the type of problem, you can definitely do that. If you want, like if you have 2 images, you want to find some sort of assignment, or identity detection between these 2 images. You have like 2 point clouds, you want to match them. The segmentation, although we are using this information about the atlas, but this assumption can be relaxed, and then we can try to just go ahead and without a prior information, just do the segmentation in the image without detecting what the identity of the neurons are. And this is just a general segmentation method, and we showed that it works better than many of the existing segmentation methods. So this is also applicable to other model organisms. And finally, signal demixing. So I think there is a lot of one of the limiting factors of current signal demixing approaches is that they cannot deal with the motion. So you need a method to actually fix the tissue before and do some sort of motion correction before running the matrix factorization models on for demixing calcium signal. But here we are relaxing that assumption, and we are assuming that there is some sort of deformation, that we are capturing the parameters of the deformation at the same time that we are actually reading out the signals. So yes, this is very much applicable to other model organisms depending on the type of problem.
That sounds really good. Do you have any support or documentation to use it to modify these softwares, or are you planning to, for general use?
Yeah, I think what we have been trying to do was to kind of develop very clean and very structured codes, and for many of the applications, the codes are like very structured. So I don't know, like maybe I can just give you an example of just go through my GitHub page. So this is we have been using like object-oriented programming, so the codes are very structured. There's a demo file for each of the codes that there's a simulation, so you simulate data and then you go ahead and run your segmentation. But then like there are different ways for calling the function of the segmentation. You can actually relax all the assumptions. You can remove the prior from that. So yeah, like there is some documentation in the codes that are on my website. But if anyone is interested in a specific use of these methods, then they can definitely reach out to me, and I'll be more than happy to help with different helping with different applications of these methods.
Yeah, that sounds good. Thank you very much.
Thank you.
And if you have any more questions, please reach out to Amin through his Twitters, which shared in the chat, or he will be around here throughout the sessions. Please feel free to post your questions. He will be happy to answer for the details and other concerns. Now we will move to our next speaker. And thank you again, Amin. It is, I really enjoy this kind of open softwares because they make the neuroscience more applicable to general use, especially for this kind of complex data sets.
Thank you.
And for our second